Good evening, everybody, and welcome to BrisScience for July 2021. We had hoped, of course, to be back in person this month at the Edge, but I'm still very excited to be here once again virtually and welcome you all to BrisScience, Brisbane's free series of public lectures on science held once a month and brought to you by the University of Queensland. I'm your host for this evening, Joel Gilmore. And I'd like to start by acknowledging the traditional owners of the lands on which we're meeting tonight and pay my respects to elders both past and present. We're going to introduce our speaker in a moment. Very exciting guest tonight. Couple of bits of housekeeping. First is, of course, tonight we are using the Zoom platform, which means that if you want to chat to us, you can use the chat function at the bottom of your screen or somewhere in your app. If you want to ask questions at the end of tonight, then you'll need to use the Q&A button. And that will go through as many of those questions as we can at the end of the evening. So chat to us using chat, questions using Q&A. I think that's everything we need to say to get started. So tonight, I'm very excited to be introducing the 2020 Australian University Teacher of the Year, Associate Professor Jack Wang. Jack has a background in science and information technology, and his PhD was at the Institute of Molecular Bioscience at University of Queensland in microbiology. But he's since applied his research teaching microbiology and training his students in both science and science communication using some large-scale citizen science projects. Everything from studying the microbes living on our bodies, which I imagine are doing quite well in the latest lockdown, or maybe that's just me, to, quite appropriately, vaccine discovery. So joining us live to send science to the citizens of Brisbane, please welcome Associate Professor Jack Wang.
Thank you, Joel. Hi, everyone. My name is Jack, as Joel has alluded to, and it's a pleasure to join everyone here on BrisScience, even though we are doing this on Zoom. I'm very used to Zoom teaching because all the classes have been switched to Zoom, and I'm going to share my screen in just a second with all of you. Okay, so hopefully you can see what's on screen. So as I said, I work at the University of Queensland, and today I'm going to be talking to you about the general concept of citizen science in my discipline, that is microbiology, and all the things I will be discussing will be present, and you can find them on my website, jackwayne.com.au. To open about discussing citizen science, we must first define the parameters of citizen science because it means different things to different contexts. And I think the easiest way to understand what citizen science is to look at the oldest known example of what citizen science could be. And that is something called the CBC, the Christmas bird count. The stories tell that the legend has it that in 1900 on Christmas Day, there was an ornithologist by the name of Chapman who decided that instead of hunting birds, they should instead count them. And this was in the Americas, where, of course, hunting is all the rage. And this began a large-scale citizen science project that still exists to this day, where people go out in the thousands, in tens of thousands, I think, volunteers all across America, counting the different types of birds within a certain sphere and using this data to contribute towards reports that's been prepared by the Environmental Protection Agency, the Climate Change Institutes. So this is a real endeavor that has an impact on genuine research outcomes in the field of conservationists and ecology. And we can use this example to extract a few key features of citizen science. So it should facilitate large-scale science participation. And this can be through scientists, although really to get it to scale, scientists are not enough. There's a lot of us, but not nearly enough to really generate the momentum we need to. You need public collaboration. And this collaboration is a very tricky thing to get right because to actually generate genuine research outcomes, the general public is not trained to the same extent as scientists are. So you have to find that good balance of the right amount of collaboration, but enough training so that they can actually contribute. And I will talk about the design principles behind this as we go today. And the last component has to be communicating these genuine research outcomes back to even more people in the general public, because the whole idea behind this is that it should be sustainable. It should continue to engage future participants, much like the Christmas bird count, which is still happening right now to this day. And this Christmas bird count is not the only example of citizen science. Many of you attending this probably are involved in citizen science projects. And of course, this is not an exhaustive list, but there's an Australian Citizen Science Association, which has a neat little project finder. And also the CSIRO has its own citizen science hub that you can find quite easily online. Not to mention most universities have some version of citizen science that its researchers are conducting. But the key thing I want to draw your attention to is that in the area of the biological sciences, there seems to be a pattern in many of these citizen science projects. So the pattern is that there's a lot of observation of the natural world because it's a very neat fit. The power of citizens and a large number of people involved from the general public is that they're spread out geographically. So if you can observe something in multiple geographical locations, if you could look at some soil in your backyard, if you can go to your nearest body of water and measure its pH, then that really is of genuine benefit to the researchers. And this natural world observation is a common template that you see in citizen science projects in the biological sciences area, although it's very quickly moving. Now, what I will say is that this is a great template for scientific participation. It's not a model that traditionally has worked that well in the university sector as it relates to my discipline of the biological sciences. And there's some evidence for this, there's some data. So within the university sector, of course, we train in science and train in research through a pretty structured hierarchical pathway. In Australia, most bachelor degrees in science go for 3 years. And if you enjoy this process, then you'll move on to further research training through honors or masters or PhD. Now, during your undergraduate studies, you may have the opportunity of doing research internships, research scholarships. And I think many of you, and I certainly was in the case, that I was lucky to get these opportunities. But if you tally up all the students that you see in our first year classrooms, and at UQ, that's 1,200 students in our first year biology classes, the statistics are pretty terrible, actually. I think on average, it's about 2 to 3% of these students actually get any research experience at all. Not that they end up in research as a career path, just get exposed to any research whatsoever. Now, this is a little dated. I think the statistics are much better in this day and age, but it reflects how difficult it is. And I'll give you a bit more reasoning why that is for more of the molecular biosciences part of biological sciences to give this accessible research experience. And of course, if you don't get exposure to research during your undergraduate studies, then you very much won't go on to further research training. And you'd have heard this in the news media about the brain drain that happens in Australia. A lot of people who are very bright are choosing not to go into science or are just not aware that scientific research is a viable pathway. And so there's lots of different factors that we can try and do to promote this uptake into research. And of course, citizen science is just one example of a way that we can allow this to happen or foster the likelihood of this happening. And that's what I'll spend the rest of the day talking to you about. The idea of getting access to research before you fully become a research scientist is not new, though. In academia, we love to name things and rename things and call it our own. So citizen science is not quite the same thing, but in academia, it's been called undergraduate research or inquiry-based learning for decades. So the idea is that it's not you sit down in a classroom and then you listen to someone tell you everything and then you become an amazing scientist. You learn by doing, you learn by problem solving, you learn by driving your own inquiry and doing your own research. And in the last few years, this term of CURE, Course Integrated Undergraduate Research Experience, has become more and more common, CURE. And it kind of has elements of citizen science embedded within it. And the key here is that students, as part of their normal coursework, hence course integrated, they don't have to go find this opportunity themselves. They don't have to email the right professor at the right time and have the magical elements aligned for them to get that opportunity. In their 1,000 student courses, they've got exposure to research, and they play a role in experimental design and driving their own investigation, which is part of learning that allows them to become research scientists. But already those of you who are in science and have done some science research training yourself, you're already thinking, this sounds like it is very prone to pitfalls, because in our area, the training hurdle is very steep. There's a very steep learning curve. So the complexity really that we can allow 1,000 first-year students to do some research on dangerous bacteria, that is quite limiting, because it's just not responsible or maybe even a good learning experience to throw first-year students right into the deep end without some good support. And so in the literature, there is this what we call continuum of autonomy and responsibility as it relates to this kind of teaching. So you can go a cookbook style where you give them a set formula to follow, you give them 10 things to do, which are all relevant to your research, and they just do it. They follow your instructions very carefully. It's a cookbook style. It's somewhat used derogatorily, although I think it has its value. They don't have much autonomy or responsibility, but they're still involved in the research in some respects. And then all the way on the other side is a research internship where they get no constraints other than maybe time. They get to do whatever they like on the topic. Of course, I don't think that's particularly feasible in big classes. So research internship is the gold standard. We want to offer one-on-one supervision, hopefully a year-long project at least to do whatever they want to investigate. But it's really more in the middle that citizen science projects would fall. Either guided inquiry, you give them some semblance of autonomy, but not infinite amount of choice. And that is the space I'm most interested in. How do we replicate this pattern of giving students enough choice that it's manageable from the teaching perspective, but at the same time giving students that sense of what a research scientist actually does? And to give you this example in another way, I've got on screen two protocols from two different fields. And there are, of course, barriers to entry. And the reason why we can't let 1,000 first-year students just start doing any kind of research in microbiology as they like. On the left-hand side is a very, very typical, quite simple in the field DNA extraction protocol. You get a bit of DNA and you try and purify it. But as you're reading through it, if you are in the field of microbiology, you'll be like, this is probably a very simple protocol. But if you're not from the field of microbiology, then there seems to be quite a lot of jargon. There's a bit of mathematics. There are solution names and names of equipment that, unless you've seen it before, you've got no contextual clues for how to engage with this. And if I sent this to a member of the general public, if I even send this to my mum, who is very educated but not a microbiologist, she wouldn't know where to start. Whereas if we use the CBC as another continued example, this kind of makes sense, right? So you've got a list of bird species you can observe. Maybe you can use an app to check and verify the visual appearance of the bird compared to what it should be. That kind of makes sense. And the amount of training you need to do a DNA extraction protocol properly is vastly different than the amount of training or the types of training you would do for a Christmas bird counting exercise. Right? Not to say that one is more trivial than the other, but the safety concerns, the additional equipment that you might need, it's very much traditionally been viewed as a barrier to entry. But we managed to move around a few of these barriers. I'm going to talk to you about, I think, 3 or 4 different citizen science projects. Not to say this is suitable for everyone's context. We've been doing it at UQ for about 5 to 10 years. And this will continue to evolve. And I'm very keen to talk to anyone who's interested in how this might work in your own context. So the first project is the Human Microbiome Project. And I think the word microbiome 5 years ago didn't have much traction in the news media, but I think most of you probably would have heard of the term microbiome by now. In case you haven't, it is loosely defined as all of the microorganisms living on whatever. So in this case, humans. So it could be all the bacteria living on humans, but it could be specific to dogs or cats. It could be anything. And microbiome just means all the bacteria or all the microbes living on that thing. And this is a relatively new research area as it goes. For the longest time, we were swabbing different parts of our body and seeing what grows. That's not a new thing. But with the advent of high throughput DNA sequencing technology, we're able to get much more resolution about exactly how much and which types of bacteria are growing at different parts of the world. And it could be in the environment, it could be in different soil samples, water samples, and indeed that's where it was first applied. And now we're looking at it through the disease lens, where we're looking at the types of bacteria living on humans, whether they be healthy humans or humans with a certain risk predisposition to certain kinds of diseases. And these projects have 2 key factors, at least the early versions of these projects. So the first thing they found was that it's actually quite interesting that across person to person, the types of bacteria they were seeing were quite different. So the term that first emerged was quite sexy sounding. It was called a microbial fingerprint, I think they got carried away in the early going as it relates to this research project. So they call it the microbial fingerprint, and it's the untapped gold mine of research data, where for the longest time we thought the human cells are the ones responsible for our fate as it relates to our genetics. But there are all these bacterial cells on the human body that have just a strong, if not stronger, role in our overall well-being. And so the race was on to find out what lived on the human body. And it turned out that across the first set of people they analyzed, the bacteria growing on them was actually quite different. Okay. And the other thing is that the bacteria growing on different body sites was also vastly different. We've known this for a long time as microbiologists, but through these DNA sequencing techniques, we've got much more information about that than ever before. So that's the first thing. Person to person, there seems to be quite a lot of variability, right? So there seems to be potential for research innovation. The second feature of these projects was that, unfortunately, at that time, the DNA sequencing technology was fast, but it was still very expensive. So the participant pool size in most of these publications were like 5 to 10 humans, 5 to 10 participants. That was considered enough. But of course, if we want to go and say that there's a lot of variability, then that is nowhere near enough people to really credibly say this is a good snapshot of what human microbiome should look like. And that's where having a big class, while originally viewed as a disadvantage as it relates to participating in research, became our greatest strength, because in our course in second year at UQ, microbiology and immunology, we have between 400 to 450 students every semester. And we went through and applied for human ethics approval to do this research. And we were able to recruit these students, hundreds of students every semester, to participate in this exercise where they're swabbing different parts of their body sites. So you can see here, most students chose, they had a choice. Most students chose the mouth, because the oral microbiome is actually very rich, and it's probably the dirtiest part of your body outside of maybe your bum in terms of the bacteria growing there. But they could also choose the ear, the nose, and the scalp. All of these are valid research questions, certainly at the time, in terms of people didn't really know how different the bacteria growing in your ear really would be from your scalp. We did do gut also for a couple of years, and that sounds kind of as gross as you would imagine. So students would go home and do a poop and then swab the toilet paper. And that was also something we did for a few years, no longer doing that. And I'll preface this with saying all of this was done pre-COVID. So oral swabs now would be unimaginable. We still have to workshop how we get this working in the current climate. So they would swab their body sites. And then with the swab, there's a few options that it can go. So we can grow the swab by swabbing it onto different agar plates, the classical approach. And there's as many agar plates as you can imagine. Different colors connote different growing conditions. And you can also do biochemical testing, look at it under a microscope, see if it bubbles. All of these are classical tests you can use to identify the bacteria. While at the same time, we had industry partners that allowed us to sequence all the DNA that's extracted from these swabs. So we didn't extract human DNA. It's only bacterial DNA we're interested in. And also all of the samples were de-identified. So they're given a unique ID. So the students know what their own unique ID is. So when the results come back, it's shared with everyone. And the students would know which one is theirs, but no one else would. They would compare the two data sets using the classical identification technique as well as the DNA sequencing technique that has the best resolution. And this is a real interesting learning outcome, because our very, very hardworking pathology labs and clinics, especially right now, they have a real dilemma because they are all about throughput. They need to get as many samples identified as quickly as possible, as we can attest to right now. And so they're very much use the best technology that they have available and go and do this DNA sequencing, do the quickest high throughput machine-based identity that we can get. But when things break down, when things are at capacity, when the machine stops working momentarily, you have to resort back to the classical techniques. And they're not really sure how to train the graduates coming in, because 99% of the time, the graduates will be using these new machines. But really, when things break down, they still need the classical techniques to identify the bugs. And so I think it's really incumbent on us as the training provider in universities to give the students that training when they're still a pre-graduation, I guess. And the last thing I'll talk about with this is that I get a question a lot about what we identify from the students and if there's anything nasty. So we've done this for about 5 to 6 years, and we've not once identified a super dangerous pathogen. And there's a couple of reasons for that. I think most of these students are roughly the same age and they're all pretty healthy. So it was pretty unlikely that we were going to find a bacteria that was super dangerous. And we never actually grow the bacteria or enrich for it. We just look at its DNA mostly. So it's very, very minimal risk. But still, we've never found a crazy pathogen in all these years of swabbing and sequencing and culturing, from students anyway. Now, the other part of this is that the research has actually moved along in this field, where originally it was everyone's got their own unique microbial fingerprint. The more that they've tested, we now know that actually person to person, the bacterial species present don't actually vary that much. But what does vary is the proportion of certain bacterial species in relation to other bacterial species. So it's not what's present, it's how much or how prevalent they can be in comparison to another bacteria, and also what chemicals and molecules these bacteria are secreting often has more of an impact than the bacteria itself. So this really has allowed the research to move along, and indeed this participation of the students. So you can imagine scaling 400 to 450 students over 5 years, thousands of participants. Their data has really gone into genuine research projects that our collaborators at the Australian Centre for Ecogenomics have been able to make use of and make real breakthroughs in this area. That's the first citizen science project, and hopefully you can see a few design elements in there. So hopefully we use the big class size to our advantage. We find a certain activity that can be trained and can be repeated iteratively, that the students can manage. Still very valuable skills to learn, but nothing too advanced, nothing too relying on independent student autonomy to figure out, and something that connects to real research. And so the next citizen science project I'll talk to you about is something that happens in third year. So these students would have already experienced the microbiome citizen science project in second year. And as they move on to third year, this is a more advanced molecular microbiology course. And the project is based on vaccine antigen design. Now, this is not vaccine against COVID. This is a vaccine against a specific type of infection that's caused by uropathogenic E. coli, affectionately referred to as Upec. So what does Upec cause? So Upec causes the nasty burning sensation when you wee, at the mildest form of it, to blood in your urine every time you stand up. And of course, that causes urinary tract infections. Now, urinary tract infections, or UTIs, happen a lot in the community, but where they impact most is in hospitals, in healthcare settings. And there's a few reasons for this. First of all, the people going to hospital, they already have another condition that they go into the hospital with. Very rarely do you go into hospital for a urinary tract infection. So usually these are maybe elderly patients going in and they're getting a hip replacement, undergoing some kind of surgery where their mobility is limited, and they're stuck in the hospital bed for long stretches of time. And of course, they can't get up and use the bathroom. So what they do is they get a catheter. And the catheter is inserted into urethra, and they wee, and it goes in a bag. But you can imagine, and I'm sure many of you have done this in the audience, if you're in any way associated with medical training, this is a bit of an awkward process to negotiate with the patients. And often, unfortunately, it's a bit of a hazing ritual in hospitals with junior doctors and the like. And so this process of navigating the catheter into the patient's nether regions is very uncomfortable, and too often it's done too quickly and not clean enough, that the point of entry is not done aseptically, so to speak, and there's a bit of contamination. And it then the problem is compounded by not being willing to change the catheter frequently enough, because the first time was so awkward already. And so it's left there to fester, and biofilms form on the catheter. And this really leads to an ascending infection from the urethra all the way up. And it could cause infection of the bladder, but it could go all the way up and cause kidney failure at its most severe. And these patients are already immune compromised, off in the hospital. And the estimate is, I think in the US alone, it's $2 billion of healthcare costs of taxpayers' money, if healthcare is covered by taxpayers, each year alone. So this is a very expensive problem. And the other part of the problem is that this infection doesn't go away if you treat it with antibiotics. It tends to keep on coming back. And the most sustainable approach moving forward is to be vaccinated in some respects against urinary tract infections, so that your immune system takes care of the infection automatically without relying on antibiotics. And the research project here is that we want the students to have the ability to help us identify and characterize some targets for vaccine antigens. And the research project goes like this. We first use bioinformatics to identify all the strains of E. coli that we can get our hands on. So each of these circles is the genomic sequence of a strain of E. coli. So blue, yellow, and green, these are all different E. coli strains. And some of them are quite pathogenic and they cause urinary tract infections. Others are perfectly harmless. And we line them up so that anytime there's a gap present in the circle, it means that this gene that corresponds on this position of the yellow circle is absent from the blue strain of E. coli. And so the logic behind this is that you want genes that are only found in the pathogenic strain and completely absent from the healthy, harmless strains. Those are most likely going to be genes that make proteins that are involved in the infection. So if it's present both in the pathogenic and the harmless strain, then that's probably just a gene that's involved in just regulatory functions, not actually in the infection. And through the work of some scientists we collaborate with, we curated a list of a couple of hundred genes that are only found in pathogenic strains of E. coli and missing from healthy strains of E. coli. This is a process called subtractive reverse vaccinology. Now, what to do with this couple of hundred genes in our list? So what will normally happen is that the scientists working on it would spend a couple of weeks cloning all of these, the DNA of all of these genes into a plasmid. And the plasmid then allows us to work with the DNA of the gene, make it into protein, put it into bacteria, and do all that work. But out of those 200 genes, we didn't really know which ones were more likely to be involved in the infection. All of them met the same criteria, right? So for a scientist to invest the time doing this, it probably wasn't the top of their priority when they've got other leads straight away. So this let us slot in very nicely because there was a real need and demand for student help. So in our course in third year, we've got 100 to 150 students every semester, and students are working in pairs, and we give them one of these candidate genes, and they have to essentially do that work that a scientist would normally do in the span of a couple of weeks to a month for that gene. But of course, students take a little longer to do it, right? So students probably take 2 months to do it, and they're only working on one gene as opposed to a scientist spending a couple of months on 200 genes. And they do the work of isolating the DNA of this gene, putting it inside a plasmid, and then expressing it into protein. It's not difficult to do, but it's time consuming when you scale it up to a couple of hundred candidates. But if you do the maths, 100 to 150 students per semester, they're doing it in pairs. Conservatively, you can go through and process 50 genes every semester and continually add to the stockpile of reagents that the research scientist has access to when they get around to actually doing this work, finding the possible candidates for the next vaccine. All right. Now, is this something that's the most efficient use of a scientist's time? Maybe, depending on the urgency, but certainly it's very good value for money for our students because they're not only involved in this real component of research, they've got some element of choice because they are choosing a gene that really comes from a giant list of 200, each with different hypothetical predicted functionality. And this is a first component of any kind of research project in the molecular biosciences, working with DNA, isolating it, putting it into a form that you can actually use and characterize it further on. And this is just some additional information about how we use the genes. So essentially, we get the DNA, we put it inside a plasmid, then we run various characterization assays to check the banding size of the DNA, the banding size of the protein, and also its presence in some kind of assay where you're looking at bacterial cell growth. All right. And over the course of the 5 or so years that we've been doing these projects, we've run many, many student surveys. And that's really the focus of a lot of the work I do in science education. So we can measure their learning outcomes, measure their confidence in specific skills before and after being exposed to these citizen science projects. And consistently, we see these confidence scores go up. But just to orientate yourself around these graphs, so essentially they're asked to agree with these statements on a 0 to 6 scale. So strongly agree all the way down to strongly disagree. And in blue, this is before they started the citizen science projects, and in red is their response after they completed the citizen science projects. And on the left-hand side is the microbiome project in second year, and on the left, sorry, and on the right is the vaccine discovery project I just described in third year. And if you break it down, what we want is for these projects and engagement in these citizen science projects to have an impact on what they think about science, right? And what's interesting is that in second year, the first block of questions asks about essentially how interested they are in the field. And second year, the pre-post testing, we're not really making that much of a dent. Okay, so where there's an asterisk, it indicates that there's some statistical significance in increase in the student responses in how much they agree with these statements. The lab experiments related to the things they're familiar with their real life. But in terms of making them more motivated or interested, it's pretty much a wash in second year. But as they progress to third year, they're really having a second bite at the apple. And you can see that consistently there's an uptick in their motivation, their interests in third year as it relates to doing this kind of work and the impact that it has on their interest in research overall. And this is further seen. The similar trend is in the bottom box here. So the bottom box of questions really asks us about career in science, interested in further training, interested in PhD. Again, in second year, we're not making too much of a dent. Pre-post testing is pretty much similar, but by the time they've had 2 goes at it, experiencing second year citizen science research, then going to citizen science research in third year, now we're seeing very clear increases in their appreciation for scientists, interest in career for science, and further research training. So I think this goes to show that as much as we like one project to make all the difference, there needs to be a concerted effort in making sure that students cannot finish and graduate their program without multiple experiences and exposure to research that then will give them a real awareness of what research actually is in the field and give them that option and pathway moving forward. Okay, so I've got about 20 minutes left worth of time of talking, I think, and I think I've left enough time for questions as well. So I'm going to talk to you about a couple of other citizen science projects, and I'm going to speed up a little bit so that you kind of get the design pattern at this point. So there's 2 other projects I want to talk to you about. Both of these are third years, so it's a little bit more advanced. So this is another project that I was involved in where there was compound library screening for drugs against tuberculosis, with the main hook here being that tuberculosis is actually a really difficult disease to treat. It's a respiratory illness caused by bacteria, and there hasn't been a novel anti-tuberculosis drug found in decades. If you're infected with TB, you just cross your fingers that it's not a resistant strain, because there are, I think, only 4 to 6 drugs that actually work at all for resistant TB, and you need to take them for months and not years. So this is a real focus in the research area to find new drugs. And what we were able to do is engineer a very close relative to the bacteria that causes TB. So its relatively harmless relative is Mycobacterium stigmatis. And we engineered a strain of stigmatis to emit fluorescence as part of its growth, so that the more it grows, the greener or the brighter the fluorescence signal is going to be. And then we asked the students to add all sorts of different drugs that were part of a novel compound library. So they screened over 10,000 compounds in the last few years and checked how much of these compounds and which types of these compounds reproducibly reduced the growth of this particular type of bacteria. And through this process, we found a dozen candidates that really are genuine compounds that might pose the future of novel anti-TB therapy. And again, it's that same design element, right? Part of the research that we do, we can iteratively repeat it, give the students hands-on with a large number of repetitive but relevant reagents that they have to work with, and in doing so contribute genuine research outcomes for the researchers involved. The next project, and this is the last project I'll talk about, is the most complex one by far. So I'm not going to belabor you with the details, but it's called beer and biofuels. And the essence of this is working with yeast and checking what type of experiment will work best that will lead to production of either beer, so alcohol, or ethanol in the form of biofuels, right? So the research questions, there could be lots of different research questions students could ask, and I think they had a choice of 3. This is one of the ones that they were given. So the presence of multiple yeast strain affect alcohol production. So again, that's super technical. We won't go into that, but they're provided with a lot of different reagents here, yeast strains, analytical techniques, equipment, and they're asked to design their own experiments from scratch and use any one of a multitude of experimental techniques to answer their questions. And you can see on the next screen that they've got a lot of choice. So they can do molecular biology, microbiology, analytical chemistry, or computational bioinformatics. And the key here is that these are across multiple disciplines And we didn't expect, and indeed we didn't find, any student who was a master of all of these techniques, because it just doesn't align that well. So what we really encouraged this to be is a collaborative group activity of 3 or 4 students, ideally all with different majors, a chemistry major, a biochemistry major, a microbiology major, and they can bring each of their expertise to bear to collaborate in answering this research question. And this is probably the most complex example I've got of a citizen science project that really still leverages the class size, but really also leverages the diversity of experience in our student cohort at that point in time to bring this complex research project and derive some genuine research answers. Which, of course, is a nice segue into what level of complexity is acceptable within a citizen science research project. And this is something I've been grappling with in the last couple of years, because I've genuinely felt that the citizen science project we're doing are of great value to the students who do them. But at the same time, I do feel the responsibility to equip them with these skills instead of just throwing them into the deep end. And any researcher knows that you can't just learn through a couple of weeks or a month. Research and research skills training takes a lifetime. All of us are still learning new skills on a day-to-day basis. And there are lots of different pieces of the puzzle that can be put together to form a fully fledged researcher. But I've just tackled 2 parts of skill sets in my area: science communication and laboratory skills. I'm going to talk about both of these in a second, but I'll begin with science communication. The way that I've decided to facilitate my students' learning of science communication is through assessment, because assessment drives learning. And this is very closely tied to the microbiome project I described earlier. So they do both at the same time. This is a task. They are given the data that were extracted from themselves. So there were 139 mouth swabs and 66 gut swabs. So this is the year where we did swab the toilet paper. And they had to communicate the findings to 2 different types of audiences, because of course, science communication or communication at all is audience dependent. And this graph that you see here is designed for someone like me, designed for a scientist. You see all the different types of bacteria on the y-axis. You see their relative proportion, relative presence. There's some statistics. That is designed for me, and they have to create that graph very accurately. But at the same time, that graph will mean relatively nothing to someone who is not a microbiologist or not a scientist. So they then have to create a short podcast that is designed to communicate to the general audience, the general public, and we ban them from using technical jargon, has to be conversational, has to be a lay language, and most importantly, it should be completed in pairs. And we ran this last year as well, and of course last year there were students who were doing our course overseas, while also students doing it locally on campus, and this was an opportunity for them to pair up and talk about microbiome, talk about microbiology across different geographical contexts. And I'm sure many of you listen to podcasts. Very few podcasts are actually just a person monologuing. Most of them are a conversation between people. So this really is authentic to the genre. Right. So this is how we really facilitated them to think about science communication. And we had an assessment task revolving around this. Now, that's within the classroom. What about in the professional context? So through my collaborations in the Professional Society of Microbiologists, the biggest one in Australia is ASM, Australian Society for Microbiology. In 2014, I launched the Communication Ambassador Program, which is a national Australian program where we give the social media channels and all of the marketing power of the society over to specific early career scientists that are working in different areas of microbiology. We give them a bit of communication training. So that's part of the package. And in return, they get control of our social media channels for 1 or 2 weeks at a time. They do a blog. They can launch whatever efforts they like. In fact, last year, some of them launched a recruitment effort to help with diagnostic reagents for COVID-19 testing. So that was a big part of that ambassador program's national profile. And it's been quite successful over time, because you can see here, this is the graph I presented when I finished this project last year, where you can see when the ambassador program launched, the society had relatively low online user impressions, but as soon as we launched, hundreds of thousands of impressions, not from one single user, because that's how it began. It was just me going and tweeting away, but it was very authentic. It was collaborative. It was a rotating curation model, and all of these early career scientists had their chance to learn their skills and apply their science communication training directly to the general public through the arm of a professional society that also supports them. And today, over 100 early career scientists from all across Australia, as well as our international partners, have collaborated in this scheme. If you're in any way related to microbiology, even if you're not, you can just Google ASM Communication Ambassador and you can express your interest. I think there's opening rounds every single year. And we also publish a paper on it in case you're interested in finding out more about that program. So that's the communication skill side of things. And to finish up, I'm going to talk a little bit about what I've been focused on most recently, which is hands-on laboratory skills in the time of COVID, when the in-person experience is no longer as possible as it once was. Now, let's flash back to a time when hands-on training and in-person was very much possible. We were assessing hands-on skills in microbiology using something we call the prac exam. Now, in our field, it's pretty common to expect students will be able to do a certain number of skills that allow them to work very safely with pathogens, looking at them under a microscope, streaking them onto a culture plate and not licking the plate or spilling it. So these are some basic foundational competencies, and we can distill them into 9 tasks that they do. We give it to them like a driving exam at the end of the semester. They do it under exam settings, not because we're jerks and want to make it stressful, but because we want to simulate the urgency of a real outbreak. Exam is not an outbreak, although I'm sure many students would attest that it's maybe more stressful than certain outbreaks. And we didn't do this by ourselves. I designed this with industry as well as 5 Australian universities that I've collaborated with. And when we first ran it in 2018, the feedback, not much positive, unfortunately. So the students said that it made it worthwhile. This is the only good feedback I could find in 2018. But even the positive, the pro, alluded to the stress behind it. And of course, all the negative comments related to how stressful this assessment item was. They thought it was ridiculously stressful, maybe the most stressful thing ever. Again, this was pre-COVID and they were doing this in person. So what we thought we would do is maybe make some online laboratory videos to help I guess, demystify what the laboratory is. And so I'm going to try and show you an excerpt of this video now. It's about 40 seconds long. So I'll see if this works. Hopefully the sound works for us. Biological organisms are constantly interacting with the environment around them. Aseptic technique is how scientists prevent contamination and the spread of pathogens. And we will cover its use in isolating microbes, transferring biological liquids, and how to avoid some common mistakes. Think about aseptic technique as moving a sample from point A to point B without introducing any contaminants into the mix. When pouring nutrient media from one bottle to another, or isolating a pathogen onto plates, contamination can come from the bottles, the plates, the surrounding airflow, the bench surface, or simply your hands. Aseptic technique is all about minimizing exposure to as many sources of contamination as possible. All right, so you get the idea behind that, right? So to get to see a video and have someone explain the theory behind it. But remember that this was still pre-COVID, so they had access to the in-person training as well. And we could use analytics to see what their viewing pattern was and get some statistics. Now, the statistics might be interesting to you or not, but I think what is interesting is the viewing pattern at the time really synced up with the in-person contact, right? So these were completely optional. They didn't have to make use of the videos, and by they, I mean the students, but you can see the viewing spikes in week 4 and in week 11. Week 4 is when the in-person lab training began that semester, and week 11 is when we ran the assessment. So they were using it in a just-in-time kind of way. They weren't watching it repetitively, although you can see the statistics here. They really were engaging with it. I didn't expect them to. And we had, I think, every student watched at least one video, I think. And those who did watch the video were watching it most of the way through, which I think is one of the more important metrics if you upload videos through YouTube, is how quickly that viewership drops off. Hopefully you're all still online. Hopefully we haven't dropped off yet today. So this is just a quick summary. I'm almost at the end of the talk, but the student feedback in 2019 very organically switched, and the stress and the consequence of the prac exam all of a sudden flipped just because they had access to these online resources. And instead of commenting on the consequence and the stress as a negative, it was being flagged as being the best part of the course. So they were able to be tested on their abilities, being able to practice a task with greater consequence. I've never really heard a student say they like doing things that are more important up until this particular comment. And I had one student, I wouldn't actually personally recommend doing this, but they decided to culture something that their daughter grew up. That's never a good idea, but it at least inspired and motivated the student to do some more work on microbiology. And I think the key here is that these online resources improve the perceived accessibility of scientific training in the laboratory. And this was for students who were on campus or had the option of on campus, but really this is something that served us really well during COVID, during the time when students could not have access to any laboratory space. And this is a project that I'm moving on and working on the next few years very, very actively. So at the moment, we are in the middle of planning a season 3 rollout. We've got a bank of a couple of, I think, 10 or 20 videos. So the channel is called Biolab Collective on YouTube. You can go look at it. And really, I'm very interested not just in the videos that I want to make, but the videos that will be of most use to the most number of people, whether it be your own interest or if you're a teacher as well and you're interested in teaching techniques or using a video as the starting point for conversation before you run the class in class or remotely. So we want to do more laboratory techniques, and I want to release more videos that are designed around ECRs, not just undergraduate students, because a lot of us know that ECRs, early career scientists, are still wanting to learn new experimental techniques. You don't want to seem dumb asking about a technique that you've never done before. So if there's a video that will be useful, or you don't have access to the equipment, but you can watch it in a video first, that'll be very great. And this is a project that's been funded by the Australian Council of Deans of Science, as you can see here. So this QR code takes you to a link that will allow you to directly tell me what videos you think would be useful. But if you didn't want to follow the link, you can also just email me or go to my website, jackwayne.com.au, for more information. This is something that is funded until the end of next year. So we're very keen to make as many videos that are as useful for as many laboratory classes and learning resources as possible. Okay, so Joel, you'll be glad to hear I'm on time. I think I've left plenty of time for questions. So I'd like to thank my project team and my funding sources, and of course, all of you in attendance. I'm very happy to stay online to answer any questions you've posed during this time. Thank you.
Fantastic. Thank you so much, Jack. That was a lot of really exciting research. It makes me wish I was an undergrad again. But we have some time now for questions. So don't forget, you can use the Q&A function at the bottom of your screen. I'm starting to see some questions coming through there now. In the meantime, BrisScience, we are hoping to be back again next month at the Edge in person, where we had hoped to see Jack this month. So that will be on the 2nd of August. Make sure you sign up to our mailing list, websites, social medias, so that you get that notification, because you will need to reserve a ticket. And of course, this video and all the others we've done are available on YouTube. And this one will go up in about a week or so. So you'll be able to catch all of this again if you missed anything or would like to get some more details. Okay, so we've got a few questions here. Jack, let's start with maybe a little bit of background. How did you make the decision to transition to focusing on education research?
So that's a great question, and a question I'm asked a lot by early career scientists. So in my PhD, I was very much research focused, like most PhD students are, but I had the opportunity, given that I was doing research at a university, to have a lot of teaching exposure. And I think to me, that was the role that I could make the most impact in after I did my first postdoc. So I applied, like all postdocs have to, for anything and everything. And this role was something that I found to give me the most room to grow in the communication skills area, in addition to the research skills area. I also apply for anything. You know, after a PhD, the first thing you want to do is like never see science ever again, right? So that's the thing you want to do. But I applied for roles in the public sector, in the private sector, different postdocs, as well as this current role that's more education focused. And to me, at the end of the day, it let me impact the most number of people on a day-to-day basis. And it gave me the room to learn all these new skills that I was just spent the past hour talking to you about. So that's what made the decision for me.
Yeah, fantastic. Got a question here. Could citizen science help us fight COVID-19? Now, this is obviously a mire that you may or may not want to wade into, but any thoughts?
Well, I think we are all participating in an enormous citizen science project that we're hoping would end any day now, to be honest. But just by getting tested, really, I mean, how much more citizen science does it need to be? But I guess even that is probably quite difficult for many people who are maybe skeptical or reluctant. So just by engaging with the science on a day-to-day basis, that is way more than I ever could have thought possible, to be honest, 12 months ago. And just the press conferences and the complexity of the language that Jeanette Young in Queensland, for example, is just using, talking about, genome sequencing as a common parlance, talking about R naught, and this jargon that's just permeated into our culture, I think is one of the very few positives that have come out of the past 12 to 18 months. Now, whether it will help us solve it is a different issue. I think still, at the end of the day, it needs to be the scientists who are working in the laboratories on therapies. But really, for us to make this and manage the outcomes of this outbreak has to be community driven. So I think we are already doing citizen science for COVID-19.
Right. Great answer. Adam asks, he says, thanks for a great talk. And he's looking forward to taking MICR 2000 this coming semester. So good that you're already tuning in, Adam. But Adam asks, is there any scope for increased complexity above basic observation protocols for community science with the general public?
So I think that has also increased a lot since the bird counting exercise I keep referencing, because we can mail out kits, right? So I think it's not purely community based, but it was a project called the American Gut, so in the microbiome sphere. So they would essentially send out these testing kits. I think in the early phase, I think they launched on Kickstarter maybe, where you have to pay a nominal amount to get the kit mailed out to you. But essentially the swab and the extraction protocol that we do in class, they're expecting people to do in their own homes. So I think that's more complex than observing the things growing in your backyard. But I think that's only because the people's understanding of DNA has also appreciated a little bit. Now, in terms of more complex than that, you start running into, at least in my field, health and safety concerns. A lot of universities have been trying like do-it-yourself labs at home. And I'm just terrified. I'm working in a building full of dangerous bugs. I would never hazard that to impose it on my students to say, just do it yourself. Or even growing stuff from your own backyard makes me quite nervous. But I think as people's awareness and appetite for that increases, that's probably where it goes. And there could be kits you can buy online or in collaboration with universities that will let you do some more advanced techniques at home.
Right. You got for a couple more questions?
Sounds great.
Great. So are your colleagues jealous now you're officially the best uni teacher in the country?
Oh, okay. It's a loaded question. Is that posed by one of my colleagues? Probably. So
Professor
the reality
Smith
of it is,
wants to know. Yeah.
Professor Smith. Yeah. The reality of it is that all of my mentors have won national teaching awards. And my building is filled with very excellent people. So I don't think jealousy is, I wouldn't imagine jealousy to be on the tip of their tongues. But also I didn't open today's talk actually with this, but I like to open every talk since winning the award with a request to lower your expectations by 20%. And that usually levels the playing field a bit more. But I think the most exciting thing is opportunities like this, because we're all doing our work in our own areas, and communicating to the general public and talking to more people is the best outcome from winning the award. Getting to form more collaborations is the best perk, I think, that's come from the award.
Yeah, great. So that has opened some doors for you, and I guess a lot of recognition. But
yeah, so I won the award a few months ago, but the previous award winner is also based at UQ. So Blake McKimmey is the last award winner. So I'm going through and checking everything he's received since he's won it to make sure I get my due. But I think that's the long-term plan. Yeah.
Nice one. All right. One more question for you, which is there any citizen science project that you've wanted to do but haven't been able to yet? And maybe I'll expand that to say, if you had a big budget, is there any project you've always wanted to do?
So I think it has to be based in the discipline, right? So it has to be authentic, and it has to be something that is of real value to the research community at that present point in time. And I think the key here is sustainability, because the microbiome project we had some industry funding, but that's lapsed. So to keep that going and give the students the same experience is actually very hard. And so I think what I would love to do is to rotate in a new bug every semester for our vaccine discovery project. And so originally began with uropathogen E. coli. We then pivoted to looking at Streptococcus pneumoniae. And I think every semester we pivot to a new pathogen to allow a new laboratory to work on their bug of the week. But of course, that relies on funding and probably a lot of safety legislation we have to overcome. So that's kind of my dream. Yeah.
Fantastic. Well, thank you for such an inspiring talk. And I'm terribly envious of your students. I mean, it would have been great to have that access to actual real world contributions, even as an undergrad. So highly encourage everyone to sign up to Jack's courses, enroll and sign up now. Good time to change careers, everyone. But thank you for your time this evening and really appreciate it.
Thanks, Joel. Thanks, everyone. Good night.